Okay, now we're on to chapter 7. We're starting to get into the more, I think, more fun stuff, the more sort of ecology sections. So we've gotten through a lot of the basic language and understanding of how organisms work and how climate impacts those organisms. And now we're getting into applying those concepts in a variety of ecological contexts. The key concepts for this chapter is that life history patterns vary within and among species, and there are trade-offs between these life history traits. Organisms face different selection pressures at different life cycle stages, and reproductive patterns can be classified along several continua. So we'll look at a number of examples here, but all organisms must reproduce some way. And the way that they do that and the size and the number of their offspring or the way they reproduce varies greatly. So we'll look at that in context to how that relates to their both ecological function, adaptability, things like that. The book starts off with this example of Nemo, and they point out that they don't use this language and it's not required for the quiz, but clownfish are what's called a protandry hermaphrodite, which means they start off male and switch to female based on the distribution of the other clownfish around it. I'll give an example of the opposite here in a second, but this sort of sequential hermaphrodism does make sense in a species where fecundity, which is a measure of the number of eggs, is linked to body size. So you can be a relatively small male and produce lots of sperm, but if you're a relatively small female, you're not producing that many eggs. And so it makes sense that if you can have the largest individual in your population be female, they're going to produce the maximum number of offspring. And so that's true for clownfish. And so in real life, 2 to 6 clownfish spend their entire adult lives within one sea anemone. They're usually not related. There's a larval dispersion. So the offspring have a planktonic stage and then settle out in a different anemone. So it's not likely that your offspring will occupy the same sea anemone you do. And the largest fish is female. The next largest is the breeding male. The remaining fish are immature non-breeders. And so there's a hierarchy there based on size. As the female dies, the breeding male becomes the female, and then the next largest becomes the breeding male. Another cool example of that is California sheephead. I guess I didn't put the species name in here. So California sheephead are protogynous hermaphrodites. So gynous like gynecology, female. They do the opposite. So proto first. So first female hermaphrodites is what protogynous means. They start off life as females, and then the largest individual becomes a male. This photo of a lunker male sheephead here was actually taken by one of the divers in my graduate lab, Kristen Reiser. And male sheephead, the largest individuals are males, and they go through a pretty big metamorphosis where when we looked at clownfish, we can assume female, male, non-breeding asexual fish. Here we've got a female. Their different coloration, their body shape is different. They have much smaller mouth, smaller teeth. Then the largest individual in that population becomes a male. They get this big lump on their forehead, and they get these huge gnarly teeth. One of their main diets are sea urchin, so they're really important for minimizing sea urchin populations, which sea urchins will then graze on kelp. And it's kind of, it's a famous story of this alteration between having a lot of urchins and no kelp, having controlled urchin populations and healthy kelp forests. So these big adult males are key to keeping the kelp forest healthy because they are a primary predator on sea urchins, which are obviously spiny, kind of gnarly things to get into. And so the biggest individuals will be males. Through fishing pressure, large males have been removed. And so now females are going from female to male at a smaller size. So that does a few things. One is it reduces the number of the big giant males around there. Big giant males eat sea urchins, keep the kelp forest happy. It also means that females are changing from female to male at a smaller body size, which means that we have overall smaller females, which means there's less fecundity per female. So ideally we want big giant males and females that are just smaller than those males, so they're making lots and lots of eggs. But as fishing pressure in particular removed large males, the distribution of the size of males is decreased, and so is the size of females. So there's a dramatic ecological impact here of losing one of our primary urchin consumers, which impacts the overall health of the kelp forest, as well as reducing the fecundity of the population by having, on average, smaller females. Sheephead are they're really, really cool fish, and their impacts are largely hook and line is one big one where sports fishers can catch these. Typically, the males are more aggressive, so you're more likely to catch a male than a female. Additionally, spear fishing, which these are big fish that stand out, and they're typically not the most shy fish. They're pretty curious, so they're a pretty easy spear fishing target. There's also a live trap fishery for these, which is pretty unique. It's a bit north of Southern California, more Santa Barbara area, where they put out live traps, and the California sheephead go in there and they catch the fish alive, and they sell those in restaurants that have live fish. So certain restaurants, when you walk in, there'll be a big fish tank, and you can say, "I want that one." So they also go into that market. So an organism's life history is a record of events related to its growth, development, reproduction, and survival. This includes age and size at sexual maturity, amount and timing of reproduction, and survival and mortality rates. The life history strategy of a species is the overall pattern in average timing and nature of life history events, shaped by the way the organism divides its time and energy between growth, reproduction, and survival. So, you can think of it as, a pie chart's kind of the wrong way, but you have so much energy, and so given that energy is finite, you have to think of how to divide that energy between growth, reproduction, and survival. Within a species, individuals differ because of genetic variation, environmental conditions, or both. And then this is an example from frogs, which should be pretty obvious that there's this interesting life history where they occupy dramatically different habitats and take different shapes. And so frogs have sexual reproduction, lay fertile eggs. Those eggs develop as a tadpole. Those tadpoles can delay metamorphosis into becoming a frog, or they can go more quickly. All of that depends on food availability, environmental conditions, how fast do they grow, how large do they get before they start to reproduce, and whether or not they care for their offspring. All of that relates to their overall survivorship. Did I not? Oh, it's down there. Okay. Sorry, I had a kelp example, but that's coming up. Okay. So some life history traits are determined genetically. Natural selection favors individuals whose life history traits result in having a better chance of surviving and reproducing. Again, fitness. But all organisms face constraints and ecological trade-offs. Phenotypic plasticity. So we talked about genotype and phenotype in the prior chapter. But one genotype can produce different phenotypes under different environmental conditions. So the phenotype is the physical manifestation of the genotype, of the genetics. And the way that that phenotype is displayed can depend on environmental conditions. So ponderosa pines in cool, moist climates allocate more biomass to leaves relative to sapwood than the same species in a warmer desert, resulting in different shapes of the trees. And the desert trees have fewer leaves and to lose less water. So we talked about that via photosynthesis and the structure of plants. So we can see that here. This here. It'd be nice if they had an index here, so we can see the desert plants here have a larger trunk and diameter versus the height, and blue would be the more coastal or temperate species. Really wish they had an index in here, or a legend. So, but we can kind of figure it out, because for a given trunk size, say, let's follow 10 centimeter trunk diameter. The height of this, these are logs, so the height of this would be 2 meters, where for the same trunk diameter, a tree, ponderosa pine, living in a moist environment would have a height of 3 meters. I'll move me again here. So this is a way of showing those. So that the relationship between height, trunk diameter, and height, the actual slope of the line is similar, but basically the intercept is a bit different. So for a given trunk size, you're taller if you live in a moist climate, where for the given trunk size, you're shorter if you live in an arid environment, as well as the difference in number of leaves to help reduce loss of water through transpiration. This is a cool example of a tadpole having different life history strategies based on if they're an omnivore or a carnivore. So depending on food availability, carnivore tadpoles grow faster and metamorphose earlier, so they're favored in ephemeral ponds that dry up quickly. So if you don't have a long time to develop, maybe you go for a larger, higher quality food source, and grow faster, metamorph more quickly before that pond dries up and you die. Where omnivores grow more slowly and they're favored in ponds that last longer, and they metamorphose in a more favorable conditions and have a higher chance of survival. So being a carnivore, maybe they're more active, maybe they're moving around more, they're larger, so they have a larger chance of being preyed upon. And so where if you're an omnivore, maybe you're a bit smaller, you can feed less aggressively, maybe you've got a larger chance of survival that way. And if your pond's going to dry out quickly, the carnivore morph is going to be more advantageous because you'll metamorphose before your pond dries up. Phenotypic plasticity is often adaptive, but adaptation must be demonstrated rather than assumed. So in some instances, phenotypic plasticity may be a simple physiological response. Growth rate increases with temperature, but we really need to, by demonstrated means that having controlled experiments where we can control for factors that would generate that plasticity. And the timing of seasonal life history activities can be of critical importance. An example of a bird migrating north too early before spring has come into full effect, they might starve because of lack of food availability. Another example I gave was around when salmon hatch from the eggs. That's based on when their parents return. And so if your parents return early, or your mother returns early, and then you will hatch out at an earlier time of the year, maybe the environment's not ready yet. Maybe there's not enough food for you. And so there's some, variability in that. And long-term data sets show that many species initiate spring activities earlier in response to climate change. And so this is often called, I'm trying to look up, is it phenology? Yeah, phenology. Phenology is the study of the timing of things. And so there's been a lot of work, particularly around plants, because they're easy to record things like when they first bud or when they first fruit. The phenology is the timing of things in relationship to seasonal cycles. And so through studies of phenology, we're seeing that many species are blooming earlier. Now, this is a big issue in climate change because we might have a warmer spring, but with climate change, we also have increased climate variability. So we could have a warmer spring, plants bud, but we still have more climate variability. So we could get a first frost that would then kill all of those buds. So it's pretty complicated. For our poor friend, the snowshoe hare, they're having an issue where when they change the color of their winter coat, that their changing of when their fur changes hasn't really changed, but the timing of when there's snow on the ground has. And so even though snowfall begins later in the year, they have this issue of having their white coat on and not having snow on the ground, so it's harder for them to hide. We'll talk about a few modes of reproduction. Why did I put the kelp one in? Oh, that's not kelp. Where's my kelp? Oh, okay, it's way down here. Okay, sorry, I'm really excited about the kelp example, apparently. Modes of reproduction. So there's asexual reproduction, which can be thought of as cell division. All prokaryotes and many protists undergo asexual reproduction. Multicellular organisms can produce both sexually and asexually. So corals are one example of that, where if you think of a piece of coral, that piece of coral is thought of as a clone. And so that started as one polyp that then spread asexually through clonal reproduction. But that whole clone also undergoes sexual reproduction as well to establish new colonies. And so if we start here, you have one polyp that attaches to a substrate or rock, begins to, frequently call it budding, but undergoes asexual reproduction. Now you've got a whole coral colony that is genetically identical. They're releasing sperm or eggs into the ocean. A quick word about coral reproduction is it's generally highly timed, and in fact, most reproduction on coral reefs are generally highly timed. And so we can predict and go out exactly when we think the reefs are going to spawn. And so all corals spawn at the same time, and a lot of other organisms do as well. The idea here is predator swamping, that things that would eat the larvae, those be so much in the ocean, in the environment, that they can't eat them all. The other big advantage for timing, or the other reason for timing, is if you as an individual coral colony release your gametes, but nobody else does, you're not going to have genetic mixing. And so timing is really important for broadcast spawners to make sure that sperm and egg come into contact, as well as diffusion rates, that really they have to be quite close. This is called the allele effect, where you can have, for example, it's a big issue with white abalone, which are nearly extinct, where you've got one here on a reef, and you've got 1 kilometer, 2 kilometers, 3 kilometers away. Well, when they both spawn, those sperm and eggs are not meeting. They're effectively infertile now. So sperm and egg meet. This zygote goes through a larval stage and then settles out as a polyp. So they do both of these, and it makes sense that, so if you're a polyp and you settle, and that's a good environment, then you can keep growing and then produce offspring to go colonize new areas. If you land in a cruddy spot, then you're dead. And so, too bad. some species have what's called isogamous, which means their gametes are always the same size. Iso meaning similar, and some have anisogamy, and not iso, same, gamete, gametes. So isogamy, like just looking at that word, you should be able to figure out what it is. Iso, same, gamete, gametes. Aniso, not the same. And so other species have different gamete sizes based on nutrition. You could think of or life stage. And so this is true in a lot of fishes, that smaller females tend to have smaller eggs as well as fewer eggs. You could think of it in birds. If they have less nutrition, maybe their eggs are smaller. And so the condition of the egg is also a measure of the condition of the organism. Here, I'll have to back up and correct myself a bit. So this is actually looking at the difference between egg and sperm. So isogamy meaning eggs and sperm are the same size, where anisogamy is eggs and sperm are different sizes. Sorry, I got that a bit backwards. So anisogamy, large egg, small sperm. Isogamy, egg and sperm are the same size, and so both gametes are contributing the same amount of material, both raw material, nutrition, etc., to the offspring, where in anisogamy, the egg contains most of the nutritional material. The sperm is just providing genetic material. Sorry, I got that a bit wrong. So, but it's still iso, same, gamete, so gametes, male and female gametes. Anisogamy, sperm and egg are different sizes. So sexual reproduction has advantages and disadvantages. So an individual only transmits half of its genome to the next generation. Population growth is only half of that of asexual reproducing species. So instead of every individual in the population creating offspring, here only half of the population, the female half, is creating eggs, offspring, however they're reproducing. And recombination and chromosome assortment during meiosis can break up favorable gene combinations. And so in an asexual world, everybody's making offspring, and if you've got a really favorable genetic code, that's going to be spread more quickly, where through genetic recombination by sexual reproduction, some of those individuals are going to get that favorable code and some are not. This is an example looking at if we had maybe the same, we can think of it as the same species of individual that can either undergo sexual or asexual reproduction, where here we start off with two individuals, and here we start off with one. These two individuals make four individuals, and here this individual makes four individuals. So here we start off with a third of the individuals being asexual and 2/3 being sexual. Here we're down to half and half, and then we go down another generation, and 2/3 of them are now asexual. It's because every individual here can produce 4 offspring, where here it takes 2 individuals to produce 4 offspring. So here the males are not contributing their own offspring independently to the population. So when you have this sort of situation, it depends on the environmental context. If the environment's very favorable for one specific genotype, maybe that proliferates more quickly. But if there's a change, then maybe these individuals die out, and then these individuals have a selective advantage, and then some members of those may go to asexual reproduction. So sexual reproduction, the recombination promotes genetic variation and a better ability of populations to respond to environmental challenges. And to test this idea, some researchers looked at a nematode worm. Oh, there's a graph here, but in this case, individuals are either male or hermaphroditic. Hermaphrodites can reproduce by self-fertilization, selfing, or by mating with males, called outcrossing. Genetic manipulations have resulted in strains of the obligate outcrossers or obligate selfers, so they have to do that. In wild-type strains, outcrossing rates increased when population was exposed to a bacterial pathogen, and obligate selfers were always driven to extinction by the pathogen. Wild-type and obligate outcrossers always persisted. So we can look at that here. So this is the percentage of outcrossing. So if we start at 20%, here, and we look at these populations through time, we have our control group that's not exposed to the pathogen. The one that is exposed to the pathogen is selecting against the selfing organisms. They lack the genetic diversity to deal with that pathogen, where the sexually reproducing individuals have that ability to deal with the pathogen. And then this is the survival rate of obligate selfers with the bacteria. So the bacteria ultimately leads to their decline. So they don't have the genetic material to deal with that pathogen, where both the obligate selfers without bacteria survive, and then the wild type, the sexually reproducing individuals with that with bacteria also survive. So in this instance, the organism can switch depending on environmental conditions. If there's no bacteria, selfing might have a larger advantage, but you have reduced genetic variability in the population. There's some really cool complex life cycles where at least two stages of different body forms live in different habitats and eat different foods. And metamorphosis is the abrupt transition between larval and juvenile stages. Complex life histories are common in insects, marine invertebrates, amphibians, some fishes. The example here is given of a larvae to an adult. So we can think of many of our aquatic insects are larval forms of flies. Sea urchins have a really different larvae, and they're actually really sensitive to water quality. And so sea urchin larvae are one of the main tools used in toxicology. So you have a bunch of sea urchin larvae and you expose them to different chemicals to see what happens. At the start of this lecture this is actually a zoea, which is a larval form of a crab. It's not a Dungeness crab. I forget what it is off the top of my head, but they're really cool. Our Dungeness crabs look pretty similar, where they have these giant spikes out the front and the back. They look nothing like a crab, and these giant spikes it reduces predation. They have these big spikes, so fish can't eat them. And they get quite large. Dungeness crab zoea, or megalope is the later stages. The megalope get large enough to where you can see them with your naked eye. And so if you go down to the docks June, July, particularly in the evening, you can see them just swarming around the docks, and those are actually juvenile Dungeness crab you can see with your naked eye. Okay, where were we? We were looking at larvae. We can go back to our friend the trematode, and this parasitic trematode that has a really complex life cycle we talked about a couple chapters ago, where it requires going through a number of intermediate hosts and has totally different shapes and functions. So it needs a really complex life history. So you have to have birds, snails, and frogs in that ecosystem for it to survive. So it has this specialized stage for each host, which is a pretty complex life cycle. Many plants, algae, and protists have complex life cycles. Plants and most algae have the alternation of generations, with a multicellular diploid sporophyte, which alternates with a multicellular haploid gametophyte. So we'll look at this in context with the kelps. So this is a sugarat kelp, Saccharina, and it's similar to, we have a similar species here. This is examples from New Zealand, but we have a similar species here, as well as our Macrocystis and Neurocystis. Macrocystis is the California kelp. Neurocystis is the bull kelp. And so you have, where should we start? Let's start with the sporophyte. So you have a mature sporophyte. It's releasing male and female zoospores that then produce gametophytes. So you have a male gametophyte and a female gametophyte. So let's go back to our language here. So the multicellular diploid sporophyte alternates with the multicellular haploid gametophyte. So diploid versus haploid, so having both X and Y chromosomes and then just one set of chromosomes, haploid. So the sporophyte is diploid, producing male and female zoospores. Those zoospores then grow into basically their own organism. They grow into either a male or a female gametophyte. Those gametophytes then release sperm and egg. Those sperm and egg come in contact and start developing a sporophyte. So it's pretty interesting where you've got a sporophyte releasing male and female zoospores. Those male and female zoospores are not fertilizing to produce a new sporophyte. They're settling down to, in this case, the benthos of the ocean, bottom of the ocean, growing out. They have to survive and not get grazed on by sea urchins. And they produce sperm and egg, which then are fertilized, and that develops into a sporophyte. So that's one example of this alteration of generations. And some species have direct development, where a fertilized egg develops directly into a juvenile without passing through the larval stage. There's an example of salamanders that lack the gilled aquatic stage that lay eggs on land, where they hatch into small terrestrial juveniles. Later we'll talk about that. In relation to snails, snails have a lot of what's direct development, sometimes called crawl away young, where they'll lay an egg and then out of that egg will come a baby snail, where other snails will broadcast spawn, and so sperm and egg fertilize in the planktonic environment, drift around and settle out somewhere. They each have advantages and disadvantages, but really when we think about dispersal, direct development typically means less dispersal, particularly in air and water, where dispersal is frequently done via broadcast spawning or your fertilized gametes moving around the environment. I'll stop there and pick it up with the next section here.